What is the means of the current? The current means ABG or any angiosperm phylogenetic group is a current system of classification. These systems which character based to related to the taxonomy, the embryological characters relation to the taxonomy, anatomical character related to the taxonomy, molecular level, then also ecological character. Several characters are related to classification, classified the APG system. That APG is a current concept of angiosperm taxonomy, also called it. So then the current concept in angiosperm taxonomy, modern angiosperm taxonomy emphasizes evolutionary relationship, means phylogeny. Evolutionary means origin, and which is a primitive, which is advanced, to identify, to separated into the groups, to call the recent classification or phylogenetic recent classification. That is current concept. Then, so it's a molecular data with traditional morphological and anatomical characters. The angiosperm phylogeny group is classification system is now widely accepted and form the basis of classification flowering plants. That is called a only the flowering plant is also angiosperm plants. Then first one, the phylogenetic approach. The primary goal of the modern taxonomy is understanding the evolutionary history of angiosperms. This is the first goal of APG system of classification. It uses DNA sequences, protein structure, and other molecular evidences to determine how plants are related. The DNA base, number of chromosomes, then related chromosomes of plants are grouped into the one family. That is called this one DNA sequences. The APG system applies these phylogenetic principles to classify plants more accurately. This one. So this APG classification is, which is also APG flexible and evolving framework. It is also monophyletic groups. Then include all descendants of the common ancestors. The APG classification focus on recognition of the evolutionary lineage and also got families in order. The integration of the molecular data. What is integrations? So the molecular data plays a crucial role in clarifying relationship, especially where physical features are similar due to the convergent evolutions, which is also DNA sequencing, DNA hybridization, PCR-based methods. These are three. It is a major part. These methods provide details data for building the accuracies of the APG. Then also called but the rethinking monocards and dicards. It is also the modern concept of the classification. The wood classification dividing angiosperms into monocard and dicard. Now, the phylogenetic studies show that some dicards are more closely related to the monocard than the other dicards. And the dicard and also then monocard is also not completely separated. Eudicards means the dicards, primary dicards, then advanced monocards, which are also related. It can be dynamic nature of the taxonomy. It is also then the angiosperms taxonomy is a constantly evolved as a new molecular data becomes available. So the APG system is regularly revised and updated to the reflected the latest scientific findings. Latest means it is also current scientist opinion, current consist acceptance, suggestions based to this ensure classification stay so other outstanding of the classification. But here meaning of the then this is also the first type of the concept or correct concept is the embryological characters in plant taxonomy. What embryology means? Dicard or monocard. Which character? Embryo which have seed. Embryo develops into the seed. Seed have two cartilages. Only the two cartilages based to dicard. Then single cartilage is monocard. That is a best or important role. Two cartilages number. These cartilage studies in separate group is embryology. Embryology is the branch of botany that deals with the study of reproductive development in plants, including microsporogenesis, megasporogenesis, gametophyte development, fertilization, development of endosperm, embryo, seed codes. These are five important basic to related to the taxonomy that is called the mini embryology in relation to the plant taxonomy. So these embryological features are embryological characters are provide the important insight into evolution relationship. The only embryology major important characters to relation to the evolutionary trends, evolutionary relationship among plants groups that are used to the plant taxonomy, support and revision classifications. So the thoroughly very early botanist like Mr. and Strasburger recognize their potential by the true importance of embryological characters in taxonomy. It is also pioneers of botanist, which based on the EPG system of classification. How Mr. and Strasburger only related to the chromosome number, chromosome characters are related to the plant taxonomy. That is was the embryonic first information. The character taxonomy was emphasized by the options and German embryologist in 1931. His work are highlighted now. Reparative development traits can help resolve taxonomy problems. Reparative, also called development. The reparative development means in the DNA, the nucleotide arrangements, the nucleotide structure base to formation of the new variety, new trait, that is embryological information. Then the embryo development, then sporogenesis, also got a fertilization and endosperm. These are all qualities are major role to the relation to the embryology into taxonomy. So then the trade can help resolve the taxonomy problems. So this is also then first point. So what are the significance of a embryology characters in a plant taxonomy? What is a significant? So this significant is also embryological characters are involving a reproductive structure like gametophyte, embryo sac, embryos, endosperm, and seed development are generally stable and lens also got influenced by environmental adaptations. So these are particularly cases to morphological characters and also called miscible or any convenient revolution process. So it is also the structure of the embryo sac. This embryo sac is a major important role to dicot and monocot differentiations. That is also one aspect. It can relate it to the plant taxonomy, that is, the embryological evidence has been effectively used to the address of the taxonomy problem at nearly all levels, from families and genera, also got a species. Specific species and species name, generic name, family name, order name, all is related to the embryology characters of plants that can is called a base or current concept of the plant taxonomy. That is, these characters are helping in resolving double systematic positions. Also called this one a doubtful systematic. Then doubted systematic position, not doubtful. We see it also. Then also both characters, morphology character, embryology character, both are related to the plants to classify that is called a current concept of the classification, especially when the external morphology does not provide clear distinctions. The external morphological arrangement of the leaves, then also structure of the leaves, and also got floral buds, auxiliary buds. Then several morphology characters are not distinct to two different plants, but only embryology characters are distinct. That is major for the according to these and also Luchsinger embryological characters rare are a most useful it flower. So these flower taxonomical ranks, families, genus, and also species. So, but less effective in a higher, uh, higher levels, like orders, so subclasses and classes, which is based on the embryological characters, means the life cycle of angiospermic plants are here. So then, eminent botanists like Maheshwari and Joan have extensively studied plant embryology and compiled a list of families and types where embryological data has so supported the existing classification or suggested a new systematic placements for a previous uncertain taxa. And the embryology is a major family or major character to classification. These plant taxonomies have utilized embryological characters in various ways in the analysis of a evolution trend, in the circumscription and delimitation of the taxa, in making a decision and systematic positions. So this is also the figures which have based on the, you see, the microbial nature, and also called the testa and tegmen, the codes, then integments of the ovi, then also pollen grains shape, then also pollen grains number, also called then the number of lobes in the anthers, bilobed, next bilobed tetrathecus or a monolobed dithecus. These flowers, which are stamens, then anthers, structures, all these basic embryological characters are taxonomic significance. The embryological features offer valuable insights of plant taxonomy, especially when morphological traits are so ambiguous. Some key embryological characters are that have been very useful determine the taxonomy relations to the embryology. The type of the first you take go the 14 important character features of a embryology in relation to the taxonomy. First one is a type of the anther or tapetum. So this tapetum is a nutritive tissue which can suppress the microspore mother cell, which can undergoes and meiotical division to give rise a four tetrads of pollen grains are formed. The tapetum is the innermost layer of the anther. It will play a crucial role in a pollen development. This it may be the two type of the then also glandular tapetum and amoeboid tapetum. The glandular tapetum is cells remain intact to secrete or nutrients. Then also amoeboid is these cells are break down and also can release content of the into the locate. That is also two type of the position of a tapetum level. The type of the tapetum is often constant with families and can aid in classification. The tapetum nature, the number of the tapetum layers, which are important for the taxonomical level to be classified the plants in different. So this is also number in the arrangement of the anther locules number. So what is the anthers are here seen that the number of the pollen sacs locally and also their arrangement within the anther are also important of the characters. So then typically monothecus, dithecus. Monothecus bilobed anthers, ditheca tetralobed anthers, the two type of the anthers which can classified in digot. Then also tetrasporangiate, four loculate, but various of like a bisporangiate, two loculate also occurs in the whole taxonomic values. The type of the anther endothecium. So the endothecium is responsible for the anther adhesions, opening of the anthers. The pollen are released outside with pressure. That is also opening and adhesions. Taxonomically significant features include the cell wall thickness of the anther. It is also fibrous bonds, structure and development of the pattern of a endothelial tissues. It is also one character for the related to families. These characters are major important role to classification of the plant kingdoms in the basis of the EPG level of 4. So this mode of the microspore mother cell division, quadriparation. Then the process of which the microspores or mother cell divides into the 4 microspore varies. It is the type of the cytokinesis, mode of the division, the microspore arrangement, tetrahedral, isobilateral, linear, T-shape. This is also arrangement of the microspores. It is also measured. These patterns are often consistent within taxa and aid distinguish the groups. This is also arrangement of the plants. Then arrangement of the microspore mother cells, which can undergoes meatically to form a four applied cells. These applied cells, it is a division. In the division of the applieds, the cytokinesis, karyokinesis are based to the classification of the plant kingdoms. These pollen gain features. The important characters includes the number and position of the germ pores, and germ apertures. It is also one characters to identification. These are all different germ pores are present in the either side. This one is the exine and sterification layers of the outer wall, number of cells in the pollen grains at the time of the anthosis. So this the bicellular or tricellular. So these characters are widely used in a phrenology, pollen study, and systematics. This is a pollen grain future. It is also fifth major characters, but it is a oval and a integument characters. So these are oval, and also there is a present of the oval. It is also in the families or the flower. In the flower, you take the longitudinal section of the ovary. It can be separated of the oval. That is called the anatomical characters of the oval is different in the different type of the families. The features are considered taxonomically important. Development and structure of the integuments. Then integument, outer integument, inner integument. That is got a seed course after fertilization. So this number of integument. So unitegmic or bitegmic. Position of the ovules. Then orthotopous, anatopous, campylotopous, circinopous, and circinotropous, and also called these 5 types of ovules. The structure is also different. Then vasculation of the integuments, which many reflect evolutionary relationships. That is the oval and integument characters. It is also one character of the embryology in relationship to the plant taxonomy. That is called APG4, recent, also current system of classification. Then additional basic embryology characters of taxonomy. Then these features are studied and emphasized in the Maheshwari, Bhojwani, Bhatnagar, and also Radford. These 4 great Indian scientists who has proposed the rules and also systems. It can provide the insight for identifying the classification of plants at a various taxonomic level, especially families and generic and species. Only embryology characters are, it can base the taxonomic levels, especially families and genera and species. Then it can form the extent and nucellus. The nucellus character is, the nucellus is the tissues surrounding the embryo sac in the ovules. In the ovules, the taxonomical significance varies includes the crassinucellate ovules. This is also broad, massive nucellus. Then also tiny nucellate ovules, thin, ephemeral and nucellus. Then also presence and absence of the hyphostates. It is also thickenings of the region of Kalacha end. The Kalacha end thickenings are, it is a one character for the identification of the same phylogenetic relations to the plant taxonomy. Then persistence of a degeneration of the nucellus during the oval development. These are also 4 major characters, which are seventh and important character of relation to the embryology to plant taxonomy. It is also called the extent of the nucellus, but 8th one, origin and extent of sporogenous tissues. So these sporogenous tissues means microsporogenesis, megasporogenesis. Then megasporogenesis, during the megasporogenesis, megaspores are formation. These megaspores is called a macrospore mother cell or macrospore mother. It undergoes meiotically 4 macrospores are formation. The refers to the megaspores mother cells arise within the ovule. Important single-celled or multi-celled sporogenous tissue. That is one single cell, multi-cell. Then presence or absence of ovular wall layers. But type of the cell division in the nuclear epidermis. The 3 characters, which important for the megasporogenesis. It is a 1 character of the embryology character. Then 9th one, megasporogenesis. So the process of which the megaspore mother cell undergoes meiosis to produce the megaspores. How many number of megaspores? 4. Out of the 4, only 1 megaspore develop into the embryo sac, or also got more than 1. Then taxonomic consideration includes it can be timely of the ball formation between the megaspores. Arrangement of the megaspores in a linear T-shaped. It is a 1 character. Then position of the functioning megaspores, usually collateral or micropilar. It is also 2nd character. The type of the embryo sac development, monosporic, bisporic, tetrasporic. Only single cell which develop into the embryo sac. Then 2 cells, then 4 cells. That is a character features of the families. It is the number of nuclear divisions which are needed to form a mature embryo sac, usually 3 divisions to reach the 8 nucleotide stages. See, here is a 3 divisions. The first division, 2 cells. Second division, 4 cells. Third division, 8 cells. So these 8 cells, which can formation of the 7-celled 8 nucleated body is called embryo sac. This embryo sac is also important for the seed formation after development of the fertilization. After fertilization, this is also 9th megasporogenesis character. Then 10th one, the form and organelles of the mature embryo sac. So the features of a significance, number and arrangement of the nuclei, with 8 nucleate type of polygonum type. It is 1 character. Then presence and absence of the endosperm, ostoria. It is also then existence of the endosperm into the other tissues. The fertilization pathway is a micropilar, collageal, mesosomal. The 3 type, mesogamy, collagogamy, and also called porogamy. The 3 type of the fertilization to enter into the pollen tube into ovule. Then the time gap between pollination and fertilization. It is also, then also type of the endosperm. The endosperms are also called nourishment tissues developing embryo. Its type and development and taxonomically important. Then also got a nuclear embryo, cellular embryo, nuclear endosperm, cellular endosperm, kilobial endosperm, other type of the process. This is what is the means of nuclear means the free nuclear divisions before cell wall formation. That is nuclear endosperm. Cellular means the walls from after reach the nuclear divisions. Then helobial means intermediate type with also called a cortical or partial wall information. This also other aspects are natural food reserves, starch, oils, and proteins. Then also persistence of the disappearance of the mature seed. These are all is also 1 major character to the in relationship to the plant taxonomy. Also got a 12th seed code characters. So these seed code is also features such as a number of layers in the testa. Seed code. Testa, how many layers? 1 coat, 2 coats, cartilaginous number. Origin and structure of these layers. Then presence of specialized cells. Example, polycystation and sclerites. So these are the seed coat traits can vary across taxa and assistance to the identification of the then development of the abnormalities. It is 1 character. These are sometimes are taxonomically significant role in the plant taxonomy in relation to the embryology. Then parthenogenesis. embryo development without fertilization. It is a one character, one rule, or one feature to related of the plant taxonomy. Then apogamy. So this apogamy means embryos arises from cells other than eggs. That is also apogamy, synergies. Then adventative embryony means embryo arrays directly from new cells or indugments. It is also adventative embryony. Then last one, polyembryony, formation of the multiple embryos in a single ovule. These are all qualities. It is also got 13. Then 12th one, pseudocode. The 13 is also development of abnormalities. So these are all the important of the relationships in embryology into the taxonomy. So these qualities are major important to the or major significant role in the APG fourth group of classification. That is called a current classification system in angiosperm or current classification system in Magnoliophyta. So this is up. So what are the means of these are application of the embryological data in plantation. What is the application? It is also called a delimitation of the angiosperms. It is a one character. Then second one is the embryological features. Embryological features have been fundamental of a distinguishing characters. Enclosed ovules, one character. Then anatrapous ovules, polygonum type, double fertilization, development of the polyploid endosperms and fertilization. These features are provide the strong evidence to the monophyletic origin of angiosperms, indicating that all flowering plants likely evolved from the common ancestor of APG4th system. Then the classification into the monocot and dicot. So the division of angiosperms into the monocotyledonous, also called dicotyledonous, means the number of cotyledonous bases, one mono or two are dicots. The other supporting embryological traits such as endosperm type, structure and development of the seed coat, then type of the ovule embryo sac formation. It is also one application for the embryology in relation to the plant taxonomy. Then identification of the specific group using embryology. While general embryological characters help define broad groups, certain exception or unique embryological traits so are especially significant for identifying specific types of examples are the unusual embryo sacs, helium SEPA, then specialized endosperm formation. That is also the unique pollination, such distinct support or exchanges of existing classification, helping involving the systematic ambiguities and also provides a class about evolution relationship. These are all is also one important applications of the APG4 in relation to the plant taxonomy character. Then embryology evidence in plant taxonomy. Embryology evidence means it can provide internal structure and development traits that are highly conserved, often more reliable than external morphological features. External morphological features, then internal morphologically. This internally morphologically, it is a one characters. So clarifying the system of relations. Then also got a conforming or revised classifications. Also result in the distributed placements of different taxonomic traits or ranks. It is a one in the applications of embryology evidence in plant taxonomy, that embryological evidence below the family levels. So how many families are major, important or significant role in embryology in relation to plant taxonomy or plant systematics? That is also caryophyllales, embryological features, the taxonomical significance. So triangulated pollen, casunicellated by tegmic ovules, curved peripheral embryo peristem, such seeds. Then also taxonomic means distinct and uniform or uniform across the orders. Then also polygonales and plumbaginales, similar embryological features of caryophyllales. indicating close evolution relationships. Then also got a lysitidalis, alternate leaves, certification statements, absence of the internal phloem and unique embryology, different from materials. Then also Iricalis order, sympedalous flowers, fallen in tetrads. But here taxonomical sequence is a embryological is homogeneity support unity of the groups. So then also Gentialis order, it is also different. Helobiaceae, helobial type of endosperm. Then also Orchidaceae, so undifferentiated embryos, which is also useful to differentiate orders. That is called embryological evidences to the plant taxonomy based on the family's level. It is also embryological evidence below the family level. It is also taxa, then kappa, also got Empiraceae, and then in the Criperaceae, also Cactaceae, Cariaceae, Liliaceae, several embryological light features and taxonomy conclusions. Not what are the relations? Then taxonomical characters and embryology characters. Also, both are related or not related. Few are not related. It can also include in the same family, but that is also so Bodamus polygonum type embryo sac retain the Bodomaceae, other moved to the Australaceae. Then also got exocarpaceae means polygonum type embryo sac cellular endosperm confirm the inclusion of a Santaceae. Then also got a paganum embryological matches Liliaceae more than Brutaceae. These are all the character features of the embryological evidences characters, which are a major important role in the. So, but this is also important of the bits. What is the means of the EPG? Means the angiosperm phylogeny group.